Welcome back to the CIBM Center for Biomedical Imaging Breakfast and Science seminar series. Hope you had a great start and wish you a happy 2021. The format will be a little different than last year. We'll still be online, but the sessions will be chaired by one of the CIBM section heads, and the speakers will be one of the affiliate or associate members of the CIBM. I'm happy to have you all back and look forward to an exciting year of great talks. To kickstart this year, we have François Lazeras from the CIBM MRgFUS section, who will be introducing our first speaker, Rares Salomir, also from UNIGE. The floor is yours, François.
I can take the introduction. So I would like just to share with you the academic itinerary of Rares Salomir. Rares Salomir is a physicist and working in the Department of Radiology, but we have a very, very close contact and collaboration with him. So, I try to go on. Okay, just for you to know a little bit more his progression. Rares is, I don't know if you see this. Okay.
Yes.
I don't know how to put. Okay, Rares is Romanian. He's coming from Cluj-Napoca, and where he had his master in physics with the highest possible grade. So he was a brilliant student, and he finished up his studies by doing un diplôme d'études approfondies in France, in Joseph Fourier University, as a master. And after he moved to Bordeaux, other part of France, and for whom who are in the field of MR since a long time, like me, you know certainly Chris Moonen. This time he moved from NIH to Bordeaux, where he developed his own group, and I think Rares was one of his first PhD student, developing already at this stage. Okay, developing already at this stage the hyperthermia in vivo using ultrasound field, focus ultrasound. And after he moved to Lyon, where he conducted his own research, or still in the field of ultrasound. And then he moved in 2008 at the Department of Radiology, University of Geneva, as a maître assistant and after private docent. And since 3 years, he obtained a title of assistant professor of radiology. And now he's continued to develop his research. And he has also a very impressive track record with more than 75 publications. He has 11 active patents, and he obtained 9 competitive grants, either European or from the Swiss National Foundation, and for a total roughly of 3.6 million of Swiss francs. And right now he is on 3 running grants, 1 European and 2 from the Swiss National Foundation. He is also working as a research leader for clinical project, first with Siemens to develop a clinical HIFU system, a clinical system that were compatible or combined with the 3 Tesla system. And this project unfortunately was stopped because Siemens decided to not to go ahead with a combination of ultrasounds and MRI. Other projects that are more clinical, the GBOR, Group IRM Bloc Opératoire Rhythmology, and I think that would be interesting for you because it's one of the first system, as you see on the right, that is a 3 Tesla Siemens system that is attached to the ceiling with a rail, allowing the magnet to move from one room to another. And this system has three functions. One is a pure diagnostic imaging function in one room. And you can move this magnet to the neurosurgical suite to make a perioperative imaging to control the ablation and the success of the surgery. And the third use of this image is for cardiac and after treatment of arterial fibrillation patient. So this project is done. The system is installed for something like 1 year, and it's working in a clinical environment. And there is a new project, and I guess Rares will tell you more about that, but it's a very exciting project for us. It is from Insight Tech. It is called Exablate Neuro, and it is a project which is conducted by the neurology and the neurosurgeon, and Rares acts as a technical advisor and technical chef de projet for this ambitious installation. And it is an ultrasound system that is put in an MR system, and it is mainly to treat major tremor in Parkinson patients. And perhaps he will tell you, but we would like to extend a little bit the research for using this technique. And the CIBM will be, of course, implicated in this research. Okay, I don't want to spend further time. I think Rares is ready with his presentation.
Okay, so now we go into the scientific presentation. So this will be about MR-guided focused ultrasound application to brain, neurology. So I will go through a review of physical principles, overview of clinical status, and also a recent clinical research. So of course, this is not done here in our group, but we are expert with ultrasound, and now we'll move towards this new application. So a short introduction first to focused ultrasound. So like 2,500 years ago, Hippocrates said that if we cannot treat with drugs, then should do surgery in modern terms. If surgery could not work, then it should use the fire, so thermal therapy. And if you cannot solve the problem with thermal therapy, then it should be called incurable. Okay. Meanwhile, there are other progresses like radiation oncology, like gene therapy. But anyway, this idea of using temperature elevation is there for a while. Now, a focused ultrasound as it has been suggested in the middle of past century. Of course, it was the time of war, and was also that lacking of imaging technologies. But still, in 942, this was the very ever first device to focused ultrasound. So it uses 4 sources. It's a little bit like this X-ray devices, but it was able to induce in a monkey brain some lesion, as you can see here. So the idea is that similar to a lens which would focus the energy of a light beam, the aperture of the lens will focus in the small focal point, and this is why the temperature becomes very high. So focused ultrasound, it's ultrasound. It has the same physical nature as imaging, so it's a longitudinal mechanical wave. But due to this 2D focusing, this concentration of the beam has much higher intensities, like 10,000 to 100,000 times higher. The frequency range depends on application, so deeper should use a lower frequency. And this technology exploits both thermal and mechanical effects. So thermal is heat deposition, just heating, but there are others which is not thermal, non-thermal, like cavitation or sonoporation. So again, we have this this model of optical lens, but light has a wavelength which is much shorter, as you know, but the ultrasound has in the range of 1 millimeter, so it's not possible to get a focal spot less than wavelengths, but still this is in the range 1 to 10 millimeters. So if you look with ultrasound to cavitation, so this movie, okay, you have an artifact here we should not look to, but we look here where is the spot, and you see as a cavitation occur at the surface between a tissue and water. And you only see this activity here, and there is no other activity somewhere else, which means this is a touchless technology, so it virtually transfer energy into a focal spot. So this is why this makes it very interesting, because there is no surgery and there's no risk for infection and so on. But still, it's a lesional technique. So the lesions are irreversible. Now, if you look a little bit more into techniques, so we would like to have some flexibility, so to be able to move the focal point, and like this, we are using what is called a phased array. It's an array of multiple sources. So if we look to the physical interferences between several sources, then the physics of waves, these are complex numbers which will be summated, makes that we only create a focal spot, and around they are mostly self-destroyed, so they disappear by interference. Okay, it's not all or nothing, but still the energy at the focal point is much, much higher than around. And then it's possible to create what is called an electronic steering or a dynamic focusing. So like this, the focal point can be shifted in space while the source, the phased array, it's fixed. And this is a very elegant method, because we do not need a mechanical actuator to move the focal point. We simply give new order to the computer, and the focal point will be created in a different location. So this is very important for targeting, of course, for a volume coverage, because the tumor is in general bigger than the focal point, so we need to sweep the tumor, and also for, it's called a motion tracking. So this talk has not a part about motion tracking, because in general the neurological application, there is no motion, so the head is rigidly fixed. But for abdomen like liver, we are using this capability to track the motion and to lock on target. So this capacity of beamforming, it's something similar with what is called adaptive optics, where different mirrors, they change little bit the direction or the phase to correct for aberrations. So here comes the MR thermometry. So MRI is well known. It's used very often in clinical routine, mostly for diagnostic. But now we're going to transform this MR scanner into a big and expensive and very accurate and 3D thermometer. So actually, there is an effect of Larmor frequency dependencies on temperature. So a spin here, which is in a cold environment, would precess faster than one which is in a hot. So it's a little bit counterintuitive, but this is the way it is. It's not a problem. We just change the sign to plus to minus, and we have the measurement of temperature. So this is the way as MR would see the focal point. So here it's a collection of 10 foci, so it's 2 times 5, and at the beginning you can see they are individual, but due to something which is called a thermal diffusion, so this is like a convolution with a Gaussian in time, they will fuse together, and what we obtain, it's a uniform thermal buildup, a uniform temperature, which could be on the range of centimeter, while the focal point is only millimeter. So we can exploit this diffusional effect to obtain something more homogeneous, and this is a real MR data. You see the transducer down and the focal point which heats up. And because MR is fast and it's accurate, we can do closed loop feedback control. So we can force the temperature at each focal point to follow a predefined temperature track. So this is good if you want to stabilize the temperature and to assure the same value in each point. So with this introduction, I will move now into the main topics, which is the MR-guided focused ultrasound for neuro application. So nowadays, this technology has received FDA and CE mark in 2016. In a neuro application, it's only one target, which is called unilateral thalamotomy, and it targets the essential tremor and some form of Parkinson disease, which is called a Parkinsonian tremor. There are many other applications which are close to receive the CE mark or FDA approved, but still they are not yet there, so it's still work in progress. It can be used, the system does not require a modification, but it requires ethical approval and IRB. So it can be used for chronic neuropathic pain, depression, obsessive compulsive disorder, and ablation of tumors at some technical limitations, so small tumors. And then something which is not using the thermal effect, but the mechanical effect. This is called a reversible sonoporation and can be used to open the brain-blood barrier, which would enable targeting local drug delivery or cell therapy. And there are also many research ongoing in animal models for instance, neuromodulation, dissolution of clots, and so on. So a short introduction. So this tremor is the most common movement disorder. The most often it's what is called an essential tremor, and there are some specific medical characteristic that this gets worse with purposeful movement, lessen with rest, and in general there are no other symptoms. Now in Parkinson's disease, there are other specifications, other specific features, so akinesia, bradykinesia, and it is a tremor at rest. It exists also multiple sclerosis tremor and other uncommon etiologies, but this is not in our field of interest for ablation. So there are different treatments. So I'm happy that Pierre Burkhardt is with us, so he can in the end complete and give you a little bit more details. Me, I'm a physicist, so I only give a general outlook. So there are other pharmacological treatments, so like beta blockers, dopamine agonists, and so on. But now we are speaking about targeted treatment. So in the 50s, it has been already developed the stereotactic radiofrequency ablation thalamotomy, but it had a quite high level of complication. This was in the early 50s. In 1987, it started the thalamic DBS. So this has the advantage that is reversible, can be customized, so can be adjusted to patient, and bilateral. Later, it has been introduced the thalamotomy by gamma knife. This, I think, has at least in Switzerland, probably to have no future for some regulatory issues. And medically speaking, the lesion would appear after several weeks or months after treatment, so it's not really real time, and some difficult to manage the case after. So this is why there is a room for a new method of targeted treatment, which is our topics of today, so magnetic resonance focused ultrasound thalamotomy. So a short review of the motor cortex network. So, okay, at the first side, this looks rather complex, but it's some diagrams which are well understood by the neurologist. And actually, there are two main circuits, striatothalamo-cortex and cerebello-thalamo-cortex circuitry. And depending which one has a disability, could yield a Parkinsonian dyskinesia or Parkinsonian tremor, essential tremor. So our device is CE labeled for treatment of essential tremor and Parkinsonian tremor. So physically speaking, we have to target this subnucleus in thalamus, which is the VIM. So if we look a little bit in more detail, we have this nice diagram of the circuits. So this is the basic atlas of neuroanatomy, and what we have to remember for the technology of ultrasound, that we should be able to deliver energy in this structure with high accuracy, because if it's not very accurate, then of course it would be side effects, and it could be even permanent side effects. So if you look to the standard technology of DBS, so depending on the pathology, there are different targets. So subthalamic nucleus for rigidity PD, VIVL for essential tremor, Parkinsonian tremor, VPL, VPM for thalamic pain, and GPI for other kind of PD and primary dystonia. So this is something which is known from the development of neurology. So the people which have been developing focused ultrasound said, of course, the target should be the same because this is the medical knowledge, but now we will try to do it differently. So mostly we will target here the VI. And for this, there is now, as I mentioned, a CE mark. So, and it's even better that in Switzerland, there is an assurance, medical assurance reimbursement. So this treatment can be reimbursed up to 22,000 Swiss francs per treatment. So this is why it makes the technology viable, not only technically, medically, but also from the business plan standpoint, because otherwise they cannot penetrate the market. So if you look a quick comparison between these three techniques, so DBS, gamma knife, and MR-guided focused ultrasound, as always, there are advantages, disadvantages. Now for the focused ultrasound, what is very important, that the benefit is immediate. So if treatment goes well, the patient gets out of the MR scanner without tremor. Of course, this is something which is non-invasive. You do not need a craniotomy, no risk for infection. There is no implant. And, you know, if you put, for instance, DBS electrodes, this would interfere with the further capability to perform MRI on the patient. So patient has DBS electrodes, but after, when you require a MR for a diagnostic for other disease, it could develop a tumor or a AVC, then it's a problem to scan. So focused ultrasound, it's a treatment which delivers energy, and after the patient can be followed as any normal patient. So the advantages are listed here, as I mentioned, and still there are some adverse effects. Also, I would speak objectively a little bit later, but they, most of the time, in a very large majority, they are minor and transient. So if you would like to see an artistic view of this treatment, so of course, what you're gonna see is not a real case. It's like a movie, artistic movie, but worldwide, now there are 65 treatment centers. So just one supplier of the technology, who is Insightec, they are based in Israel. According to public data, at least 3,500 patients have been treated, and there are more than 80 peer review articles about the clinical application. So now, based on the MRI data, the neurosurgeon will define the target, and then the engineer will assure that the system executes accurately the treatment plan, and this energy goes through the skull and would hit. So now you see the temperature elevation is rather low. It was about 50 degrees. So with this temperature, there is no permanent lesion, but there is a transient effect, and then we can confirm that location is okay, and then a higher dose would create an irreversible ablation, and then the tremor is much improved. So you see some example of hand capabilities before and after. In Switzerland, there is one center which is working as a clinical routine. So the closest to us is a Sony module. This data is taken yesterday from their internet site, so it's public. And what they claim for is they have performed already 360 interventions and 470 targeted sonication. So sometimes there are repeated treatments or are 2 per session. There was never seen a bleeding. There was around 1% effects on neighboring structure, and only 2% of worsening, so which means 98% of improvement. So it's very good. I have to compare with other technique. And what is quite impressive is the targeting accuracy. So what we are speaking about is to send an ultrasound wave through the skull. So this is a quite thick bone, and due to some techniques which I'm just gonna mention in a few minutes, we can reach this sub-millimeter accuracy. So it's really impressive. So try to imagine what is a fraction of millimeter deep inside the brain. Of course, this addresses the problem of true skull sonication. So if you just take a HIFU transducer and we send a beam, of course, that the skull will induce a strong distortion. And it's something which cannot be used, of course. So with such a pattern, it's not a discussion to be used. But there exist algorithms which permit to compensate. So this is called the time reversal technique. And it's possible to pre-deform the waveform, and when it goes through the skull, it will self-focus. So if you look here to these simulations, so this is a focal point which is very fuzzy and cannot be used, but this one, it recovers its nice shape as a very small rugby balloon. So the way the scientists are doing this compensation, so they are time reversal technique, they are CT-based technique, or real-time magnetic resonance acoustic radiation force technique. So this one would be the most elegant because it's done in situ, it's real time. But of course, it takes some time. So until it is accelerated, it's not really used in clinics. So in clinics, the most often is used CT-based technique and analytical technique. So it's very fast. The refocusing quality is not the best, but it's acceptable. But it has the advantage to be readily available and not to increase the treatment time. So basically, we need the patient CT data of the skull. After there is some physical model where we calculate the distortion of the beam, and then we apply phase correction. So actually, we perform an inversion of the distortion. It's what it's called an inverse function. And in the end, the result, theoretically, we should get here a Dirac function, but in reality, it's a focal spot, which is quite accurate. Now, the question is how we do the targeting. So actually, ideally, we should be able to see on a true image where is the target, but even with a 3D system, this is not yet possible. So the targeting is done based on an atlas technique. So the 3D MR image has to be fused with the Morel atlas. And then there are some indications. So this many millimeters from ACPC line, other millimeters from the PC, and so on. So in the end, the neurosurgeon will give to the computer interface, so yes, I would like that my focal point appears here. Of course, if would have 7 Tesla data, then this would be totally different. So we are not yet there, but in the future, I think there is room for coupling this MR-guided technology with preoperative 7 Tesla image fusion, and in this case, this basal ganglia and thalamus can be explicitly shown. So until 7 Tesla will be included in the workflow, the method is based on the Atlas fusion with 3D 3 Tesla images. So now the procedural steps. So prior to the treatment day, we need some pre-treatment planning. So CT scan, of course, 3D MR. Then there is we perform a registration, and on the treatment day there is some preparation. So of course the patient head should be shaved. So this is the only, let's say, uncomfort for the patient. It's clean, and then we install a stereotactic frame. And with this, the patient would go into the helmet. And of course, there is an elastic membrane with a central opening where water would circulate. So I would not say further for MR physicists and MR engineers that there is some water leakage. This is not very good. So the system is waterproof. And then how the lesion evolves. So if we look with T2 MR images, but there is no contrast agent, so this is native. here in this case. So at immediately after thalamotomy, we have a lesion here. 1 week after has a slight tendency to grow, so probably this is some tissue swelling. And after 1 month after, it will show the final lesion. So this is one advantage, that it's possible to see the lesion just immediately. And the very first paper, which has, as far as I found, it has been published in 2013. So of course, at that time, the system had not this CE mark. So it was a research study, was on 15 patients. So of course, for ethical reason, they include a severe and medication refractory essential tremor. But nowadays, you don't need to wait until reach this point. So eligibility point could be sooner, because there is more proof this is safe. They obtained from the very beginning this initial test or trial 75 reduction in contralateral hand, with some side effects in 4 patients. We will speak a little bit later about side effects. And then there was another study which follow mostly the same principle. And they found that the tremor was abolished immediately. So again, this is the second main advantage, that if everything goes smooth, then the patient has the benefit just at the end of the session. Again, this minor to moderate side effect, which we will detail a little bit later. And nowadays, we can also perform such a system on a 1.5 Tesla system. So in hospital here at the University Hospital, the system will be installed with a 3 Tesla, because this is the gold standard. But if for some reason there is no other way, it's still possible to do on the 1.5 Tesla with a similar outcome. So there are many other publications, and the one which I'm mentioning, this is for neuropathic pain. So this is not about a tremor, but still it's a future indication for the technology. So where comes this complication? Of course, not going into too much medical detail, but just to give you an overview. So most of the time, the patient, in a few percentages of case, can report paresthesia, numbness, imbalance, or guide disturbance. But they are mild, and they should resolve over time. So of course, if we look into this very complex wiring in the motor pathway and basal ganglia, we have to target this nucleus. But there are other circuits which are nearby. So the premotor pathway, the somatosensory pathway, and the auditory pathway. So this is why this kind of side effects in general are linked to the proximity of the fibers, and this kind of side effects have been in general reported. But now, with the improvement of technology, I repeat that these side effects goes to a few percent, and is never something which is dramatic. Okay, so for the last slides, I would also mention what is done as a clinical research. So clinical research is done with the existing system, but off-label. So does not require a technical modification, but it is used for other indications which are formally not administered on the regulatory point of view, are not yet approved. So the first idea is not to do only unilateral, but to do bilateral thalamotomy. So this is a case report. So the first treatment has been done in the past. So let's say this is this one. Sorry, this is after 25 months, the image of the lesion, and this is 1 month. It means there are 2 years between the treatment. So it's not something which is done immediately, one after another. But in this case, after 24 months, because the patient outcome was very good with the first treatment, then he underwent a treatment on the other side. And then with the same principle that they delivered a small dose of energy to locate the accurate target, and then the treatment was complete with ablative sonication, and in this case they obtain so without side effects. So I think this is the best case for the patient, that now he has been treated both sides and neither had side effects. But this approach is not yet CMARC, so it's not the rule. And other applications, so now everything is where we can target the beam. So we cannot go everywhere in the skull, in the brain. So typically we cannot target yet the cortex. This is a physical problem because of the beam forming. But they could target other targets which are a source of other neurological disease. And in this study, so the target was the anterior limb of the internal capsule, and what they report was improvement in the score by 61%. So it's still a good result if there are no side effects. Then also some application for psychology, so treatment of depression. Okay, this is only a case report, but it was quite a desperate case, and they went for this ablative technique and obtain a dramatic improvement in depressive symptoms. Other clinical research is done with a 220 kilohertz applicator. So this is something which has to be added extra. So when a hospital would purchase a clinical device, then research money should be obtained to purchase this extra part. But the electronics is the same. So we only change the applicator. It's the same like an ultrasound scanner. You change the probe, but you keep the main unit. So this has something to do with the brain-blood barrier. So normally this is something to protect the central nervous system, but sometimes we would like to penetrate and to enter with drugs or even with immunocompetent cells. And for this, it's possible to apply low dose sonication, this lower frequency. And this applicator has the advantage that we can come closer to the cortex. So again, not yet the cortex, but it's much closer as compared to the deep brain, which is used for the ablative technique. So the first report about the brain barrier opening in lateral sclerosis has been published in Nature Communication like 1.5 years ago. Okay, in this study, there was not yet a treatment, but was only demonstrated that it's possible to open the brain-blood barrier to permeabilize, so for further delivery of some drugs. Other application which is seen with a lot of promises is Alzheimer's disease. So in this publication, they target the right frontal lobe. So here is the sonication pattern. So the focal point was serially placed in a 9x9 millimeter pattern. Now, why 9 and how? This is something which is, of course, decided by the physician, which is in charge with the patient. But on the engineer side, we have to guarantee that the device would execute precisely the treatment plan. And what you see here, this is a T1 with gadolinium uptake. And before treatment, of course, there is nothing, but just immediate, there is this uptake here. And then 24 hours after, there is no more uptake. So it means that this brain-blood barrier is normally closed. So this is why we are speaking about a transient effect. So you open the window, deliver the treatment, and this would naturally close by itself. So it's not something permanent. And most important in this kind of study, basically there are no reported side effects or impairment of health of mental condition, because it's low dose, low energy, and it's transient. So hopefully also we'll find here the scientific environment with the University Hospital of Geneva and CHUV and EPFL and so on to move on this also very exciting technology with BBB opening. Okay, and something similar, but in this case the target was different. So you can see the different location, and again they report very nice. So this was hippocampus, and they reported very positive results. So again, no side effect, and 95% of the targeted volume. So what has been planned to open the barrier, it has been opened really. And about oncology, so this is a little bit more complex, because in oncology we need to target a larger volume. But this study, so this group of Toronto is of course very active. So Professor Hininen, which is the most active in the field, it's a study about 5 glioma, and this was done in the course of chemotherapy. So now I cannot tell, and the author cannot tell that yes, chemotherapy was more effective. So it's not an efficacy study, but this was a feasibility study, and they targeted here the peritumoral tissue, and this peritumoral tissue is demonstrating opening of a brain-blood barrier. So logically, it should be that given that there is a chemotherapy, then a higher dose of drug has been delivered to the tumor. So now it's too early, of course, to state about the efficacy, but you see that the methodology is there. And finally, there are other studies about, this is more fundamentally, something about neuromodulation, also okay, with or without drug delivery. Of course, the system which will be purchased here at University Hospital of Geneva is not dedicated to this kind of fundamental research. For this, we have to apply for a different kind of technology, which use a dedicated transducer for a rat, so it's much smaller and more accurate. Okay, so other studies have been reported on the primate. So okay, I think I'm done. I'm mentioning also as a potential application to dissolve clots, with the risk that after there are small fragments which will migrate further, and this is something which is not yet clear, but there is a possibility to break the clots with the HIFU, but of course have to be should be taken that you do not disseminate the small fragments, and from one problem we create 5. Okay, so this was for my talk. I would like to warmly acknowledge my colleague Oran Lorton, who has helped me with the presentation, and also helped a lot with the development of focused ultrasound technology in Geneva since 5 years. And the technological information was provided or taken from the public site of InsightTech company. So with this, I'm open for your questions. Thank you.
If you have any question, please, it's time to ask Raresh. He's with me. If you see him in the...
Can I ask something? I actually have 2 questions that are linked between them. I don't know if I missed, but I didn't understood at which depth you can penetrate according to the intensity, the frequency that you can deliver now.
Yes, okay. So actually, it's mostly about the distance from the bone, from the skull. So the system cannot focus close to the bone. It should be like 5 or 6 centimeter after. So there are, of course, some diagrams, and I will send you after. So there are some diagrams, how far away is the reachable, the targetable region. So the system which is for ablation, which works at 650 kilohertz, can only work roughly around the deep brain. But the other system, which is 220 kilohertz, I think they can go something like 3 centimeters under the skull. So by now, this is a fundamental limit, and it's little chances to fast improvement because it's fundamental physics, and this is the way that ultrasound propagates. So yes, this is a geometrical limitation.
Yeah, and I get
applications. Yeah.
Yeah, thank you. And for the fact that you have limitation in reaching the cortical structure is more linked to the anatomical characterization of these structures, or what is the issue behind?
No, it's only about the bone. So the beam has to cross the bone. And in general, it's easy to do so when the beam comes perpendicular to the bone, you see. So it's something like this, that if the beam comes perpendicular, it will go, but if comes tangent, it will reflect.
Okay.
And, this is the difference in impedance between a bone and water. So of course, if you want to speak about cortical tumors, this is not very likely, but you saw that with the this low frequency applicator in image I have shown, there was some example of brain-blood barrier opening a few centimeters under the skull, the bone.
Okay, thank you.
I have a question.
Okay.
Sorry.
Go ahead.
Hey, Rares. Servus Rares. I have a question. I'm Christina. So, a very nice talk. In our lab a few years ago, maybe a few years ago, somebody tried to use ultrasounds just to break down the blood-brain barrier in animal models, just to be able, even for DNP, for dynamic nuclear polarization, to inject different polarized substances which need some time to pass the brain and go in the TCA cycle. What's the feasibility for preclinical studies? So I haven't seen any results. So yeah, I don't know why. So.
Okay, so yeah, my talk was mostly about clinical research, a clinical application, because I had to focus somehow.
Yeah.
Have this new project in Geneva, but there are still some literature, and if you look, there is one company. Okay, I'm not advertising, but I know there are not many of them in Bordeaux, and they are delivering plug and play devices. So depends on how is your MR scanner. They make it compatible with a small animal Bruker scanner, for instance, or so. And this is something which is, I mean, ready to use. So it should work. After all is in the power you apply. So if you apply, of course, too much power, this would create irreversible lesion, which is not the purpose. And if it's not enough power, then there is not opening. So the point is how you can control the amount of energy. And this is something which is, of course, individual. So even from one rat to another rat, or human to another human, of course, there is no the same power, because the skull is different and so on. So they are including something which is called a cavitation detector. So in general, when this sonoporation occurs, there are also acoustic signature where different frequency from the emitted frequency is observed. And these devices, I mean, a good quality device should have what is called a cavitation detection. And like this, you can apply the right dose. So probably if you tried at the past and didn't work, is because the amount of energy was not correct. It was too much. This would create occlusion and coagulation, and there is stop. And if it's not enough, of course, no effect would occur. But in preclinical in animal models, this is something which is currently published, and the principle is demonstrated. So now I think the focus is not if we can do or cannot do. So this is something which is a technical level, and you just buy the good system to be successful. After, now they look into the efficacy. So after they do a tumor model, and they look if a survival rate is better with or without BBB opening, with chemotherapy, and so on. So I would
not give you now publicly the link, but after I will send you by email this example of a commercially available system. So something around 100,000 francs. And with this normally, so from the experience of other sites, so they use it in USA, in Texas, Utah, also in Paris, with the group of Michael Tonter, and they are using this device and it's working. I mean, otherwise they would not publish and it would not be purchased.
Okay, thank you. Again, very nice talk.
Other question? Daniel, you had a question?
Oh, yes. Thank you for your talk. Excellent. One question regarding the focusing of ultrasound beam. I'm talking about the piezoelectric disc, for example, and I remember some papers from the past in which 64-channel applicator, HIFU applicator was developed, and to get the focusing around 15 centimeters, the size of the transducer or the piezoelectric disc was, well, diameter was around 7-8 centimeters in this particular design. I'm talking about 64 channels. And I wonder how this focusing, which is 15 centimeters, is achieved once you have 500 or 1000 channels, because then the size of a single piezoelectric element is significantly reduced. I simply wonder how it reaches so deep in this case.
Mm-hmm. Yeah, so in general, the smaller the element, the higher the range is, because we have to think as what is called a radiation diagram, yeah? So just the reciprocal principle. So if the source of a wave is small and becomes comparable to the wavelengths, then this would be roughly a spherical wave. And when it becomes larger, then it's more directive, okay? Okay. So the smaller the element, the larger the steering range. Now this system has 1000 roughly, I don't know exactly, say sometimes it's 1000, it's 1024, 980, but okay, roughly 1000 elements. And then we can calculate. So you take the surface of the helmet and we divide roughly, okay, it's not 100 padding because it's also some randomization and so, but we can have an idea how big they are. And actually, this system needs this focusing mostly to overcome the problem of the bone. So what is called beamforming, so to apply RF delays on the signals. Here it is required to compensate for the distortion of the bone. So this is why after the steering, the capability to steer after is it's reduced. If you would use the same system in free water, you have a lot of steering, but through the skull, most of the capacity of beamforming is occupied, consumed, or taken as you want by this problem of going through the skull. On the other side, even if you use very small elements and you have a theoretical large steering range, still because of the problem it just Janina mentioned before, so the reflection on the skull, we could not go very close to the skull. So in conclusion, the steering range is only a few centimeters around the deep brain, but even so would not be useful to go further because we cannot penetrate then the bone. And the system has quite a flexibility, so the elements are small and high number, but this capacity is mostly used to calculate the inverse function of the bone.
Okay, thank you. So, but when I have, let's say, a single piezoelectric element, let's say with diameter of 8 centimeters, which has focusing point, let's assume 15 centimeters, and I take the same piezoelectric disc and I reduce the diameter to, for example, 1 centimeter, it will not reach that deep, right? It will not have... Because
it's only one, but you have 1000, you see, so it's the...
Okay, so it's about beamforming, okay.
Yeah, it's the summation. So each wave, if you only activate one element, of course, you do not get focused. You get something like this, well, not a spherical wave, but anyway, it would be a clearly divergent wave. So it would be just the same as you turn on the light, and farther from light, you have less. But with many elements which are placed on the spherical cup, when you do the summation, the waves would arrive at in a coherent interference at the focal point, and they would self-annihilate, so it would be negative interference outside. So this is, yeah, how it, of course, with one element is not possible. If you want a simple system, then of course it should be the ratio between diameter and focal length roughly 1. So this is what is called the focusing number. You are right. Yeah.
Okay, thank you. May I have one last question? Because I was just wondering how the RF part of 3T system is implemented in this HIFU system you are using. You have body coil, right? And do you also have a couple of channels for receive only, or how is it done?
Yeah, so until now, the system installed, they are using the body coil because there is no place to have another coil. So the space around the head is already taken by the HIFU, yeah, or the focused ultrasound. But since this year, Insight Tech, they are providing a system with coil, MR receive only, of course, receive only coil, which is embedded in the system, and actually it's part of what they call a treatment kit. So a treatment kit, it's something which is disposable, and first reason is to assure to be waterproof and not to have water leakage between the acoustic coupling and the skin. But in this treatment kit, now they also include receive only, so it should be a RF loop, and then this is connected as a third-party coil to the system. And of course, that there is better signal because it's a surface coil. The counterpart is this is not very homogeneous, as you know. So a surface coil will give a good signal close, but it will be is losing signal in the far field. But we do not look with this for diagnostics, so we don't really be sure that the left hemisphere has the same signal as right hemisphere, not to be prone to confusion. So what this is important is the MR thermometry, and the MR thermometry is measured as a phase contrast. So even if the coil sensitivity is not uniform, is not so big problem. So of course that the accuracy will change, but the measurement is correct. So in conclusion, until last year, the MR thermometry intraoperatory has been done with the body coil, which of course is the worst coil. But now they developed this surface coil, which comes with the disposable kit. And then it's like a loop which would take here the front and the occipital part.
Okay, thank you very much. Thanks a lot.
Okay, thank you. I think we will stop the question here. Thank you, all of you, for your presence. I think it was a good talk. And I let perhaps the conclusion remark to Pina, if you have anything to add.
Yeah, so thank you once again, Rares, for the amazing talk. I think everyone really enjoyed it from the feedback already on the chat.